name is Christina Joseph, and I did my research on the effect of the DSK gastropeptide on feeding behaviors in Drosophila Me. Now you see, eating is vital for proper human functions, and eating disorders such as anorexia and obesity, malnutrition, all hinder our ability to live a proper, healthy human life. And from previous research, we know that feeding behaviors are linked to certain genes in the body. One such example is cholecystokinin, CCK for short, which is known to be connected to the digestion of lipids and proteins within mammalia. Now, we wanted to see whether upregulation of CCK would lead to the upregulation of digestion of lipids and proteins, leading to the upregulation of satiety levels within mammalia, therefore downregulating the amount of food intaken by the mammal. But you see, manipulating genetics in mammals is a really hard thing to do. So we went to the next best option. We went to Drosophila Me, which is commonly known as the fruit fly. And thankfully, there is a homologue for CCK within the Drosophila Me known as DSK, Drosophila. So we conducted 3 sets of experiments to see the effect of upregulation and downregulation on feeding behaviors within the DSK system. So the first set of experiments we did was the PER assay, also known as the proboscis extension reflex assay. We timed flies, starved flies, to see how long they would begin proboscis extension and how many times they would extend their proboscis. Because you see, when flies want to eat, they'll extend their proboscis. Now, we saw that regular flies ate for a long period of time and didn't take too long to begin eating. But you see, flies with activated DSK neurons took quite a while to begin eating, if they ate at all. Almost 67% of flies do not eat in this experiment. So we saw this qualitative data, and we wanted to make sure that flies were actually legitimately eating less than our wild-type flies. So we went to the colorimetric food assay. Here we found that flies with downregulated DSK peptide ate more than our wild type, while flies with upregulated DSK peptide ate less than our wild type, which is in accordance to our hypothesis. Now we wanted to check one more area of feeding behavior, which is known as food availability feeding. So we wanted to put flies in a low availability arena and see whether or not they would still eat, whether or not, depending on their satiety levels, which is connected to the DSK gastropeptide. We found that when we downregulated the DSK gastropeptide, there was an increase in intake of food. But we did not see the same with the upregulation of the DSK gastropeptide. This could be because of some discrepancy of the genetics, and we need to conduct a whole new round of experiments. But all in all, the data shows some really promising results. And based on this data, if this data is replicated in our second round of experiments, then we can move on to a mammalian model, rats, and if it's replicated in the mammalian model, then we can truly say that CCK has some effect on feeding behaviors in the circuit. Thank you.